So thank you so much, Rosemary, for joining us and welcoming our speaker today.
Yeah. Thank you. Hi, everyone. It is my great pleasure to introduce our speaker today, Filipa Rijo-Ferreira. She is a new assistant professor of infectious diseases and vaccinology at UC Berkeley, where her lab studies circadian rhythms in parasitic diseases such as sleeping sickness and malaria. In these systems, the host, the parasite, and the environment all have daily and seasonal cycles, and the interplay between these is fascinating. She's published on this work in journals such as Nature and Science. Dr. Rijo-Ferreira received her MSc in 2009 in molecular genetics and biomedicine from the Imperial College London. She then completed her PhD in 2016 at the University of Porto in basic and applied biology, molecular parasitology, and neuroscience. Following that, she did a postdoc in Joe Takahashi's circadian lab at UT Southwestern, and as of just this year has started her own lab as a new assistant professor at Berkeley. I first met her at a conference in Uruguay several years ago and have heard her give several talks since, so I know we are in for a real treat today. So without further ado, I will hand the microphone over.
Thank you so much. That just puts a lot of pressure. Well, actually, I am sorry, but now I cannot share my screen anymore.
Sorry, you should be able to share it now.
Okay, now I can. Yes, thank you. Wonderful. So yes, it's really a pleasure to be here. And just like we've discussed, if anyone wants to interrupt, I understand that the audience is broad, which is very exciting for us to have different perspectives. So if it's not clear anything, let me know. I would rather prefer everyone to understand. So we'll be focusing today mostly on circadian rhythms in parasites with a focus on malaria, but we'll also touch a little bit on other parasitic diseases. Malaria, it's caused by a parasite of the species Plasmodium, but it's transmitted by this vector mosquito, Anopheles species. So it's a female mosquito bites, and if the female mosquito is infected, it will also deliver parasites during a blood meal. And despite all the efforts on the malaria community to really eradicate these infections, unfortunately, we are at the steady state of these 500,000 deaths every year, and we are not being able to bring that down. And unfortunately, most of these deaths are in children under the age of 5. So many different efforts and different perspectives are still welcome to try to understand how we can further bring these numbers down. From my perspective, I was very interested in malaria, not only because of its health burden, but because it's been very famous for periodic fevers. So malaria has been known for these fevers even before we knew it was caused by a parasite. So over millennials, there are descriptions of a disease that had fevers that would come and go at certain times of the day. And now we know this is a consequence of the parasite replication cycle in the blood. So I have here a schematic for us to see. So this is a really amazing phenomenon because you can imagine that this is a single cell parasite. It's coordinated with other parasites in the blood. They are invading the red blood cell, then they divide and then burst the red blood cell at a particular time. And people have shown that if you put these parasites in culture and synchronize for this replication cycle, the messages, so the RNA that these parasites are expressing is rhythmic too, meaning that our genes that are messages that are on and off depending on the time of the day. And you can see here this heat map exactly showing genes like that. So each row would be a gene that it's either on and then off, or it's off at the beginning and on at the end. There are many other rhythms described for these parasites, but one thing that is interesting is that depending on malaria species, this cycle can take 24 hours, 48 hours, or 72 hours. So modulo of 24, which was very interesting for me. What's special about these 24 hours is that organisms like us and other kingdoms have evolved a way to anticipate the rhythmic environment of our world, our 24-hour light and darkness, high temperature, low temperature type of cycles. And these circadian rhythms, in mammals are quite understood and well studied. And the sleep-wake cycle in particular, which is controlled by these, is regulated by an area in the brain, in the hypothalamus, called the suprachiasmatic nucleus. This is a small 20,000 neuron type of nucleus that really can define the timing in our body. All these other dots here that I highlighted, these are different sleep promoting areas or wakefulness promoting areas, but the timing itself is mostly regulated by this suprachiasmatic nucleus. Inside these neurons and virtually all other cells of our body, there is a molecular clock. So at the molecular level, how this takes 24 hours can be simplified by a core loop. So you can think of transcription factors as activators. So there's two of them, clock and BMAL, that bind to the E box, so the area that regulates the transcription of genes. And then the genes, when they bind, get transcribed. And among the genes that they transcribe are their own repressors, cryptochrome and period. These are the repressors that can then translocate back into the nucleus and compete for this binding, so no more transcription starts happening. And this takes 24 hours to be completed. The activators can also bind to many other promoter areas and lead to the rhythmic transcription of genes involved in metabolism, in the immune response, in redox. And that's how the circadian clock really controls many physiological aspects, not just the sleep-wake cycle. So in the case of malaria, the periodic fevers have always been assumed that were driven exactly by this circadian rhythm of the host. We have such clear circadian rhythm that could be what is underlying these malaria fevers. But there is an experiment that really put this hypothesis into the mainstream that I thought it was a really beautiful experiment, and it's done in the 1930s, so a long time ago. So these researchers were studying monkeys, and they were testing the importance of light shifts and jet lag in the malaria infection. So if we follow these wiggly things as sleep of the monkey, these representations, so here will be a night, so the monkey is sleeping, and then it's awake, and then the next night sleeping again, and so on. And these monkeys were then exposed to a 12-hour light shift, meaning that would be like us traveling 12-hour time zone, potentially like from here, from California, where I am, to somewhere in Europe. And what happened is that these monkeys would experience jet lag for a few days, and then read again this new light and dark environment and get synchronized to match again their sleep to the night. And what these researchers were actually following were the parasites inside these monkeys. So they were following a particular stage of the parasite cycle, and they would see, oh, the stage is happening at this time and it's not happening on the other time. And they were following the blood stage of the parasites. And what they found was that when the monkeys were exposed to this 12-hour light shift, the parasites also experienced a moment of jet lag and then got resynchronized to the host. And that was really the basis for the common idea that the circadian rhythms of the host were driving parasite rhythmicity, which might have well be true. So they thought, okay, we have it all figured out. But I had already done some other projects in another parasite called sleeping sickness and show that even though parasites may not be really exposed at any stage of their life to the environment, they may still have intrinsic clocks. So I created the hypothesis that perhaps this was more complex, that these rhythms in malaria infection are basically a consequence of both the integration of the host rhythms, but also a malaria clock. So I set out to test this hypothesis. And the reason this could be potentially advantageous for the parasites to have their intrinsic clocks would be, instead of just being able to read the environment and adjusting to it, it will give them the possibility to anticipate the rhythmic changes. So the nutrient availability is going to be different because inside our body, we eat at particular times of the day. So you can imagine that nutrients availability is rhythmic. Also, our immune response is rhythmic. It's been shown that either the number of immune cells available or how reactive these cells are is changing throughout the day. So the parasites being able to anticipate these could be an advantage as well. And I mentioned to you that the way this infection is transmitted is through the bite of a mosquito. And so the mosquito itself also has a circadian rhythm and bites specifically at night. So a way that parasites could anticipate when this biting happens could be potentially important for their transmission. And so another possible advantage for them to have an intrinsic way to tell time. From our perspective, why should we care? Why bother finding this out? Well, I think we should care to understand the world around us. That's for sure just a fundamental question of biology. But we can also think that if parasites have circadian rhythms and their biology is different throughout the day, that means that maybe the drug is more effective at certain times of the day too, the drugs that we use to kill them. We can also think that if they have a rhythm that is important for their biology and establish an infection, if now we can disrupt that intrinsic clock, we may be tilting the scale towards the host. And so I'll touch a little bit on both of these later on. Okay, so how can we then dissect if there are contributions to the parasite rhythms beyond the host? So the monkey experiment had light-dark shifts. So is it really light playing a role? And so we could try to address that by eliminating light from the equation. So we set two groups of mice, experimental mice. Some were in light and dark normal conditions, and some mice were in complete darkness. What happens is because mice have their intrinsic rhythms. So if we can think about these in terms of a sleep-wake cycle, in the case here, these wiggly things will represent activity. So mice will be active, so not sleeping at night because they are nocturnal. And we had these two groups of mice, and then we infected them, and we waited a few days and started taking samples from the blood to assess what's going on in terms of the parasite transcriptome, meaning the RNA messages that are being expressed at certain times of the day. So we first took unbiased approach with multidimensional scaling to see how the overall gene expression of these parasites are at these sampling times. So we found something interesting and important, that both the time points, the biological replicates, so the parasites taken from different pools of mice, they were clustering together if they were taking at 16 hours. So that's good. They were also clustering together independently, whether it was from the dark, complete darkness group, or wild type, both were wild type, or light and dark. So that didn't matter either. And another aspect that we saw clustered together was that the 16 hours were clustering with the 40 hours and with the 64 hours, which are all collected 24 hours apart. So these already told us that it seems that the overall gene expression of the parasite seems to show this circular pattern, because things are changing and they are coming back to the starting point 24 hours later, which was something very exciting for us. When we then actively search for circadian rhythms, meaning using algorithms that really look for cycling of genes, you can see here the algorithms feeding the raw data underneath. We were able to then quantify how many genes were cycling. And we found that in both these conditions, light dark and complete darkness, the parasites had about 80% of their transcripts being rhythmically expressed. And here you can see that this is heat maps showing the up and down regulation of genes. And the order is the same, so you can see that the profiles are virtually the same. So these led us to conclude that the light itself is not really driving, it's not contributing to the rhythms of the parasite. So what was the next question, was to try to see how thermodynamically locked are the parasites to a 24-hour world. And that is actually a characteristic known for other circadian systems, that not only you can entrain, synchronize to new environmental cues, like in jet lag, but also if there is an environment that is slightly different from 24 hours, but within the physiological range, organisms can adjust. And that was a good way for us to see if this is really something common for parasites. Will they stay locked for 24 hours, or are they also going to be able to readjust? And we took advantage of a mutant mouse that has a long period because it lacks one of the components of the clock. What happens is that now the clock of this mutant mouse has a long period of 26 hours. So it wakes up later 2 hours each day. So here it's the way we plot the activity recording, so the behavior of these mice. We have a cage with a wheel, and we can monitor when the animal is active. And so here on the y-axis, you have multiple days of recording, and on the x-axis, you have a higher resolution of 24 hours plotted twice. So here in the black bar is the night when mice are active, and here all these little blips is running wheel revolutions. So you can see that these mice show an active period at night, and they're resting, almost no activity during the day. And this is in a normal light dark condition, but then we release them here on this shaded part, is complete darkness for many, many days. And you can see that they still maintain an active period and a resting period. The same for the long period mutants. However, in this complete darkness, they will rely only on their clock, and because their clock is 26 hours and not 24, they wake up 2 hours later each day. So this was a really good setup for us to see, because if we wait 5 to 6 days, now these two groups of mice, they are basically at the opposite phases. Whenever ones are waking up, the others are already going to sleep. And so we could test what happens to the malaria parasites. Are they going to maintain the wild type timing, or are they going to be now shifting to a 12-hour later phase? And when we look at the gene expression of the malaria parasites, you can see that by the total number of genes peaking at a certain time, they are not overlapping. So there is already a clear difference in the phase. It seems that they are slowing down their rhythm. And if we now align each gene to, instead of the actual clock, to the intrinsic clock of the host, we are able to basically see that the profile is maintained, which is here compared each gene in the phase is expressing each gene of the 4,000 genes. When in a wild type mouse or in a long period mutant, they do have a correlation, as you can see here. However, you can also notice that it's not a perfect fit on the red line here, which tells us that even though they are adjusting to a new environment, they are not being simply responsive to it, but they are actively trying to adjust, which for us was already a little hint that maybe this is really something that parasites are actively trying to do with their intrinsic clock. And we'll come back to that. Third question. Okay.
There's a question.
Yes.
There's a question from Luis asking, in what way do you use the term thermodynamically?
Well, that's a good question. So here we thought that since it's such a clear 24-hour window that parasites take to complete their cycle, or a 48-hour or a 72-hour, depending on the species, it had been proposed that just that's as long as it takes to complete the type of things that they need to take. It's just how long it takes for the DNA to divide, the membranes to create, and it was just how the system is. There's not really intrinsic ability to that. It was just as the locked thermodynamically, meaning that biologically, all the things that need to happen at this particular time, because they have no other way to do it. That was how we address that. It was actually a term proposed before us. That's how the system works. I hope that explains what I mean.
Thanks.
Okay, great. Thank you for asking questions. And so, okay, third question. One proposal before us was that in more depth, not only the parasites were being driven by the host's cue, but the host rhythms, but in particular the feeding rhythms of the host. And that has been proposed because parasites do seem to sense it and adjust their timing also to match the feeding. And so a way that we came up to test if really the feeding rhythms were driving such parasite oscillations was taking advantage of an automated feeder system we developed in the lab that basically allows us to now abolish feeding rhythms by making food available evenly distributed throughout the day. So if we have mice in this particular situation, the activity, so the running wheel, will still maintain at the nighttime. However, the food that they normally eat at night in this spread out, evenly distributed feeding starts becoming constant throughout the day. And here is the same graph as I showed you before, but now overlaying also in red the tiny pellets that mice take. And so in a normal situation, you can see that both the wheel activity and the feeding is happening at night. However, if now mice are not allowed to take food whenever they want, but only whenever it's available, which is spread evenly, you can see that they still prefer to run and be active during the nighttime, and their food instead is being taken throughout the day. So this is a way to also split these two behaviors that are usually together, and also to abolish the feeding rhythms by themselves. So then allows us to test how necessary are these feeding rhythms for the rhythms of malaria parasites. And what we found was that the replication cycle under the microscope still maintained rhythmic. And regarding the gene expression of the parasite, whenever we look also at whose genes are cycling at which time, we saw that independently, whether there are feeding rhythms or not, malaria parasites still show the same profile, really concluding that feeding rhythms by themselves, they are not really required for the parasite rhythmicity, even though parasites may be able to sense them. But there are many other rhythms going on in the host. And so a way that we took to approach that, other rhythms that we were not able to abolish when you abolish feeding rhythms, like the activity rhythms, the temperature rhythms, was to take advantage of another mutant mice that now lacks two of the repressors, the cryptochrome 1 and 2. And these mice become arrhythmic once they stop having environmental cues to rely on. And that's what you can see here on these behavior plots. So again, the wild type animals we've seen, so when in complete darkness, which is all this shaded area, is still a very clear active period and a rest period for the feeding and also for the activity. But in these arrhythmic mutant mice, they show a clear night active day resting period if light is present. But as soon as lights go off, you can see that they become randomly active or sleeping. And that's the arrhythmic aspect that I'm referring to. So that's how we can take these mice and infect them once they are in complete darkness with malaria parasites, and see if parasites themselves become arrhythmic similarly like the host, because they were solely relying on host cues to become rhythmic. And so what we found with the first multidimensional scaling approach that I showed you before, so good things right away, there is a circular pattern that's promising. And then we start actively looking for the rhythmic gene expression to quantify. And we found that even though there's not 80% of the genes, like in a wild type healthy mouse profile, cycling in the malaria parasites, there are still 60% of malaria genes that are rhythmically expressed even when the host has no rhythmic cues, really proposing that malaria parasite rhythmicity is not driven by the host, but intrinsically generated by the parasite. We can think now that what happened to these 20% of genes that stopped cycling. And I think there are potentially two components to that. So maybe some of them were really just responding to rhythmic host cues, and some of them are probably just becoming below our detection limit. So remember here, we are measuring the RNA expression at the populational level of all the parasites. If now parasites are slowly drifting apart, they will stop being less synchronized, and so we will stop being able to detect all the rhythmic genes. And that is actually something very well understood, for example, for mammalian cells. So mammalian cells have intrinsic clocks, and when they are in an environment that is rhythmic, with receiving rhythmic cues, then as a population, you'll see very beautiful high amplitude rhythms. However, once they are released into constant conditions, so no more rhythmic cues, because each cell does not have a precise 24 hours. Imagine some will have a 24-hour 0.1 or 23-hour 0.9. Slowly, as time passes, the population starts drifting apart. And here, this is an experimental data supporting that. So you can, here as a tissue, there's communication between cells. If we would follow the expression of once a day of a clock gene, which is what we are measuring here, you can see that is once a day the expression, and all cells are doing the same. Whereas if now you dissociate this tissue into culture and you follow neurons, and there's no synchronizing cues available, what happens is that everything becomes more like a Christmas light all over the place. But if you follow each single neuron, which is here represented, you can see that a particular cell has a very clear circadian expression of these clock gene. And so at the populational level, if there's no synchronizing cue, at some point you have each cell with its own phase, and so the average is just becomes flat. And so there's this damping happening during the population. So we thought that maybe a way to test that out would be to follow what happens to malaria parasites as time goes by of having no environmental cues, no host cues. So this is basically recreating the same experiment as before. So after this is starting to plot data from 5 days after the infection, but mice have been in constant darkness for over 2 weeks at this point. And if you follow a particular stage of the parasite in a wild type mice, you have the presence of parasites at a particular stage, no absence, so present, absence once a day. And in a rhythmic mutant mice, we see that as well in the first few days, but as time goes by, at the populational level, you start having a lack of synchrony, really suggesting, similar to what has been described to other circadian systems, that you also still need synchronizing cues at the populational level. So parasites are not able to self-synchronize among each other. That's what this experiment tells us. So we thought a good way to complement these wet lab experimental data would be to do some computational mathematical modeling. And that was all driven by John Abel, who has been an amazing collaborator, which I tried to bring him to me today, but unfortunately he's on well-deserved vacations. So I'll try my best to describe what he did. So John created 4 differential equations to try to model each of our hypotheses. Is the parasite rhythms driven by the host, driven by the clock of the parasite? Is the parasite driving its own clock, but is it relying on feeding cues of the host, or is it relying in activity related cues of the host? So trying to model that, and then try to challenge these equations to replicate similar experimental setups that we did. So suddenly I lost my mouse, I cannot point. So on the top plot, you can see that there's the yellow wiggly thing is representing the feeding profile of the mouse. In blue, you have the mouse activity rhythms. And in pink, the light pink, you have actually 100 lines overlaying because he's modeling 100 parasites. And in the dash red is the populational level, what we can measure at the RNA sequencing, all the sequencing results I showed you. So in a normal wild-type host, you can see everything is cycling. On the middle panel, now the host has this arrhythmic feeding, so this spread out feeding regimen. And the model that is really testing whether it's intrinsic for the parasite and it's relying on activity rhythms to get synchronized didn't become affected, similar to what we observed experimentally. And finally, if the parasite is exposed to a rhythmic feeding and a rhythmic activity of the host, the population of parasites will slowly drift apart. And so you can see that the dashed red line is now also starting to damp. So this is the one that's better represents our experimental observations. And so this is really a model that also supports the idea that malaria has an intrinsic clock, and it's mostly relying on activity cues of the mouse to get synchronized. And here I show you also the results of the other modeling, that you can see that the parasites lose their cycling right away if there would be feeding driven, and they also would lose rhythmicity right away in the last panel once activity is removed, the activity rhythms. And the feeding being entrained, being the most reliable cue that parasites would be sensing, it's also not really representing what we saw experimentally, because you would expect a decline in amplitude once they were abolished, and that was not what we saw. So altogether, we seem to favor that malaria indeed has an intrinsic clock and mostly relies, if present, on activity-related cues of the host. So that's exactly the main conclusions of this study, but these really raise many other questions, and that's things that I want to follow up now in the lab. How exactly are the parasites sensing the host? What are the most important cues? Try to create a hierarchy map for those cues. And if indeed these are intrinsic to the parasite, how are the parasites having an intrinsic clock? So what are the clock genes are the next big questions. And the reason that's not such an easy task is because even though circadian rhythms I mentioned to you are conserved across the tree of life, even bacteria has them, animals do share the molecular components that regulate the activators and the repressors. However, plants, fungi, bacteria, they all have their own. So there seems to be a conversion evolution of the molecular players. And so where would these protists, malaria parasites, single cell parasites lie will be somewhere here in between these two, and we'll need to figure out what are their own molecular components. But one thing that is shared is the importance of having these clocks. And that is independently of being a plant or being a mouse. If you disrupt the circadian clock of these organisms, there's decrease in survival, and in the particular case of mammals, there's also known for metabolic syndrome. There's a higher incidence of cancer. So altogether, circadian clocks really seem to increase the fitness of organisms. So we can think that that's also going to be shared for parasites. So if we are to disrupt, we are potentially going to make their very good establishment of infections weaker, and potentially also affect the way that they are able to transmit diseases. And so finally, on the last 1 or 2 minutes, I just want to bring to your attention the part of the circadian medicine for infectious diseases. So I briefly mentioned that one particular aspect that could be important for us to think about rhythms in parasites, or actually in rhythms in pathogens, could be because there might be a window of opportunity to when we treat. And this has been all started with another parasitic infection called sleeping sickness, which has a very beautiful, also single cell parasite with that swims around in the blood, so it's extracellular. And this sleeping sickness, just for a brief context, it's also an infectious disease, of course, parasitic disease, but it's unlike malaria that it's spread all over the world. This is particularly common on the African continent. And the number of cases has been really dramatically reduced, mostly because the vector, so that tsetse fly, is not as skill of a vector as the mosquito, so it has been easier to control. But unfortunately, because it's such a rural type of infection, it's believed that most cases actually remain unreported. And because unlike malaria, this parasite is not picky on the type of host. So it infects humans, but it also infects wild animals and cattle, creating a massive economical burden. It's estimated about $5 billion each year to this continent in terms of cost consequences for this infection. So there's still definitely a relevance to studying it. And for what gives name to the disease is the sleep aspect being disrupted. So in a normal healthy individual, we know that by our own experience, our sleep is condensed during the nighttime. But here you have the profile of a sleeping sickness patient. And you can see that among other aspects of the sleep architecture, the timing is very obvious, which was what piqued our interest in terms of circadian rhythms in parasites. And we actually had another story that I'm not going to mention today about sleeping sickness being definitely not just a sleep disorder, but a circadian disorder. So what I want to tell you is that sleeping sickness also has a clock, similar type of experimental data, but in culture, so nothing other than the parasite, no environmental cues, and there are still 1,000 genes of this parasite being rhythmically expressed. And for particularly this parasite, it seems to regulate the Trypanosoma brucei, the sleeping sickness parasite. The clock seems to regulate its metabolism. And you can see here, if we measure just energy as ATP molecules, we can see that the metabolism is changing in the parasite. There's two major times that it happens. And this was very curious because if we know that many of the anti-parasitic drugs actually act on metabolic pathways, perhaps then they're going to be more efficient at different times of the day. And so that's exactly what we tested. We started different dose response curves, meaning an increasingly higher dose of a medication that's used in the clinic to treat this infection called suramin. But we started this treatment every 4 hours through the day. And we found that there are shifts, meaning that it matters when you are initiating the treatment because you can have over 2.5 times the need of a higher dose to kill exactly the same number of parasites. So there are indeed differences in the susceptibility to treatment depending on the time. And so this is something we've been thinking about, time of day administration, for example, for management of cholesterol. So people usually take their medication at night. For high blood pressure, usually that's higher incidence in the morning, and that's when you should be taking some of the medication. So just people have been starting to think about that, but not for infectious diseases. And so I think this is a good proof of principle that I wanted to share with you. So altogether, what the messages that I think would love for you to take out of this conversation is that parasites may have their intrinsic clocks, depending, at least for malaria and sleeping sickness, probably others too. they still sense the host and use those cues to synchronize as the population. That's where they exist. So similar like us, we rely on our light and dark cycles. They also rely on host cues. And some parasites like the sleeping sickness can disrupt the host clock. And we should investigate if there is a window of opportunity for optimizing the current treatments. For example, sleeping sickness, the treatment itself is so toxic. There is believed that 5% of people actually die of consequences of the treatment. So if we can just optimize the concentration of drug you need to treat, may make difference. And so that's it for what I had to tell you today. And I just wanted to highlight my postdoc mentor, Joe Takahashi, which has been extremely generous, my close collaborator, Victoria Costa Rodriguez, and John Abel that I told you today, and other people from other projects that I briefly mentioned. So thank you all for your time, and I'll be happy to take more questions.
That was a wonderful talk. Thank you so much. And I think
Thank you.
I think, you know, it really sort of underscores the importance of doing really well-constructed experiments and coupling them with mathematical models. That I thought that was beautiful, the way everything came together. And so I really enjoyed that. We have a few questions in the chat. So I'll start with one from Luis. He asks, what would be the impact of no activity in the mice? That is to say, what if you took the wheel out of their cage, made the cage very, very small? What would happen?
Good question. I don't know the answer, but I am thinking that potentially it's not the actual movement of the mice. However, it can be exacerbated. So I'm believing that mostly is the temperature related. So there are temperature rhythms in our body that are going on once a day as well. So for particular mice, there's about 2.5 degrees Celsius that there's a fluctuation. And I have done experiments in culture, and I know that, for example, the sleeping sickness parasites are able to sense fluctuations in temperature. So I'm believing that's one important cue that it's becoming amplified with the activity of mice. So if you're running, of course, your temperature also raises. So then the amplitude is bigger. But I'm not believing that is the actual movement. I'm thinking it's more the indirect things that it creates. So potentially, even if you were able to restrain the animals for abolishing any movement, I would imagine it would still be underlying cues that parasites would sense, and so they would still maintain the population synchronous. That's just a guess. We would need to test that out.
That makes a lot of sense. I hope it makes sense to Luis who asked the question. Kind of following up on that, you mentioned at the very beginning of your talk that malaria was known for causing these rhythmic fevers. And I'm curious whether those are driven by the parasite's rhythms or the host's immune rhythms or some combination?
Yes. So definitely, it's a very important thing that we need to figure out exactly the outcome. But I think it's a combination in a way, because it's the parasite having rhythms and bursting at a particular time and sensing the host to do so that creates this coordinated aspect of the red blood cell release. So the bursting of the red blood cell, it's what causes the fevers, because when they burst, they not only all the suddenly there's going to be tons of parasites going around, but they are also bursting red blood cells that are full of metabolic waste of the replication. And that itself then triggers immune response that creates the fever, right? So in the end, I think it's going to be a combination of both, because the synchronicity of these bursting is controlled by both.
Thanks. More questions in the chat. So Jolene asks, she says, wonderful talk. Since there's advantages for gametocyte density to peak at the same time as biting hours, do you think that there will be a shift in parasite clocks as peak biting shifts earlier in the day in response to vector control? Or would this take a really long time for us to observe a shift like that in the field?
Yeah, very informed question. Thank you for that. So yes, there has been actually already reports that because of control efforts in terms of using of bed net, the biting of the mosquitoes are being described to happen also earlier in the day when there's the window of opportunity for biting to occur. So I think the question is whether this is going to create evolutionary pressure for parasites to also shift their own availability in the blood for the transmissible form to go inside the mosquito. And if we are going to be able to see that in real time, or if this is going to take, thousands and thousands of years. I have no idea, but I know that in a very small amount of time, parasites have been under massive evolutionary pressures with drugs, and they've been able to develop mutations to avoid those very quickly. So that's in terms of resistance for drugs, it's been very, very, very strong evolutionary pressure. It could be that in this case is not going to be necessarily the need for mutations, but perhaps there are polymorphisms in malaria parasites that make some of them already naturally peaking earlier, and we're going to be selecting for them because of the way the mosquito are biting. So I don't know, but that's a really cool problem.
There is another maybe related question in the chat. Also, do you think that the parasite rhythm has an important role during the mosquito phase of its life cycle?
Yes, I thought about that a lot, and that is because if these intrinsic clocks are important, they should be important for many different aspects of parasite biology. And I thought about that a lot in terms of the mosquito as well. And so I've been trying to go and look there. And also in particular, I've been very interested in what happens to parasites in the salivary glands of the mosquito. So the last stage where they are before they bite a host, so where the parasites are ready to infect, there they've been described to be there for weeks and being dormant, waiting for this encounter. And so because they do not replicate, it also is a very good spot for us to now dissociate between replication and only clock related genes. And so I can tell you that our preliminary data shows that there are rhythms even in this dormant stage. So yes, we believe there's going to be important for their biology there too.
Luis asks, what is the light-dark change function? Is it a step? And what would happen if it were more sinusoidal or any other type of change?
Maybe you can clarify. Is it because it's asking whether it's like an on-off instead of a gradual?
I think that's what he's asking. Maybe Andy can unmute Luis.
Yes, that's wonderful talk, by the way. Yes, that was what I was asking. I remember reading somewhere else that, and maybe I read it incorrectly, that if changes that are used for entrapping the clocks are more smooth, the system seems to be more resistant in that case than when it happens as an on-off. And so I'm wondering if in some of these studies, by using kind of more gradual changes, right, that you don't go full light, full darkness, but have more that if there could be additional behaviors or additional resistance of some behaviors that could be exposed in that way. And I may be totally wrong.
No, I mean, it's a fair question. So I thought, I don't know if I've heard about that particular having sinusoidal being more resistant to change, but definitely it will be a much closer mimic of what the reality is. So it might be relevant to consider doing it. So the way our experiments were done, and also the monkey experiments, was really like light on off. But there are definitely transition for trying to also have a more gradual, like you're saying, kind of creating like dusk and dawn type of scenarios. In these particular experiments, because most of the experiments we are doing in complete darkness, might not really change much, right? Because it's complete darkness. So there's not really importance of light in darkness anymore. However, yeah, it could be that maybe what you're referring to is the ability of clocks to adjust to differences in the overall time of length of the day. So for example, mice, which is where we do a lot of experiments, because of course you can recreate all of these things, they are able to adjust to a day that it's 24 hours, clearly, like a 12-hour light and 12-hour dark. But also if now the day has 20 hours, it's still something that they can do. But if it's maybe 18 or 16, it's not, right? So maybe that's the gradual thing that you're talking about, and that's definitely something that has been something very well studied, and there's a point that the animals just ignore, so they stop adjusting to that light dark schedule.
I was, if I can just one thing, I was thinking that some of those things could be possible to check in the model, and some of them might have an impact also if you are thinking about strategies with regard to potentially giving drugs or kind of mess up with the vector, like the mosquitoes' circadian cycles and stuff like that. So it's really a fascinating topic.
Thank you. Yes, and definitely a lot of different things that we need to consider for moving forward. That's for sure. Thank you.
If people have questions, we still have time for a few more, so feel free to drop them in the Q&A. And until you do, I will take up the time by asking more questions. So I'm really fascinated by the disruption of the host's circadian rhythms in sleeping sickness. And I'm curious whether it disrupts also body temperature rhythms and other things that are thought to be under sleep and circadian control, or just the sleep behavior.
Yeah, no, that's very good. And I didn't have, of course, time to tell you much about that story, but yes, indeed, it disrupts not just the sleep-wake cycle, but it also disrupts the temperature rhythms, so the body temperature rhythms. It actually, overall, it seems that you have early in the infection, all fever type of things. So you stop having rhythms because the temperature is also very high. And then it becomes, as the host is able to control more the level of parasites through their immune system, it becomes regular temperature. But then as the infection progresses, as the mice become sick and start having more the sleep-wake cycle disruption, they have also shown some hypothermia, but with it, so temperature drop, but the rhythms themselves also stop being as rhythmic as they were before. So there's an overall effect, and it seems that in particular the way that these sleeping sickness parasites disrupt the host is by accelerating the clock. So instead of having a normal timing of things happening, everything starts happening earlier, and that's, I think, what is underlying the main symptom of sleep is that sleep occurs now during the rest phase, right? During the active phase, sorry. And so that's how potentially it's also going on in humans. So what people describe is not the overall symptomology, is not that they are overall sleepy, but they are sleeping during the daytime and they have insomnia at night. And so this possibly is because it's also to humans, the parasite is also accelerating their clock. And so everything starts happening earlier. And that's what we see in mice.
That is fascinating. Fascinating.
Yeah, thank you. We have no idea how the parasite is doing it though. And so that's something we want to understand it further. It's like, what are they secreting? What are they doing that it's making that?
Yeah, and whether that is in the parasite's interest too, right? Or whether it's...
Yeah, what was the evolutionary pressure for things to start happening this way, right? So I would imagine that accelerating the clock would not be something that would be of interest for other parasites, just because otherwise maybe it would have happened in other infections too. And so far, this is the only one that we noticed things like that happening. So generally, people have described only a reduction in the amplitude of rhythms, but not an acceleration of rhythms. And so it might be something specific for this infection.
Really, really fascinating. Well, thank you. If there are no other questions from the audience, I think we are basically right at the time limit. So, so thank you again for coming and giving the NICO seminar. Beautiful work, really fascinating story.
I'm very happy. Thank you. And thank you so much for all the good talks and good discussion. It was a pleasure.
Thank you. Thank you, Filipa. Thank you all of you for joining us today.
Thank you. Have a good day. Bye-bye.